Okay, so Dr. Kenny is here. He will give a guest lecture next Tuesday about like a protein in the muscle. So I'm going to go over the basic knowledge of a protein, like amino acid, structure, a little bit of everything. And then we will know the basic knowledge before move forward. He talks about all those details, like application in the food products. So when we talk about the protein, first of all, they come from a Greek word which is called proteast, and that means the first kind. And some of the textbooks, they say it is first importance. So that means it is very important in our life. And we talk about the function of the protein. You already learned in the other classes, biochemistry, biology, or general microbiology, then we will have like a support function. And it's a muscle tissue, it's collagen, and it's a support function. The second one, it will have a transport function. And some of them we call it a permease. And it's like a bridge, have a tunnel, and then the materials, nutrition materials can transfer from the outside into the inside of the cell membrane system. Then the number three, it is obviously we know the enzymes, most of the enzymes, or all the enzymes are protein. They are regulated the biochemical reaction. Let it happen more efficiently. Then it's could be a regulation of the body, such as like some of the hormone. For example, like insulin will be adjusted the blood concentration in our body. And the last one, we talk about the protection. And these days, you heard all the time, we talk about antibody and antigen, and especially antibodies. These all belonging to protein. And it's go on and on. We can mention lots of the function of the protein. Now, what is the single units of a protein? It is called amino acid or amino acids. Now we know that there is 20 natural amino acids. Some of the textbook also mention it's 21, so we'll talk about that. So first of all, what is the basic structure of an amino acid? So we can draw is in the middle is a C. Okay, and we call it alpha carbon. And then it's connected with the H, which are hydrogen atoms. And on the left side is NH2, we call it a amino group. On the right side, we have COOH, it's a carboxy group. And then, there's on the top, there is an R, which is a side chain. And this side chain, it decided what kind of the amino acid it is. And here, we write it like a flat, but it is actually cubic. So there is an angle here. This angle we call it's like a phi. And here the angle we call it PSI. Okay. And because this is the alpha carbon, so amino acid, it is like a chiral constructor, and most of these, or all of these natural amino acid, it is L amino acid. So here is the basic structure. Now, how we gonna be categorize the amino acid? And we say that based on the side chain R, we can categorize into different groups. basically we have is a non-polar. Non-polar, most of them cannot dissolve into water, so we call it a hydrophobic. And then we have a polar amino acids. It's all of them are hydrophilic. And the hydrophilic, they also can differentiate into two groups, actually three groups. One of them is non-charged. And the second one, and third one are charged, which is called acidic, will be negatively charged, and basic, and will be positively charged. So we're going to go over all these details of the side chain of the amino acids, and then we are going to be categorizing, and we'll talk a little bit more everything. Some of them will review we learned before in my general microbiology class. So let's talk about that. First of all, let's draw here. So we take this out. The first group is a non-polar group. So because the HNH and the COOH, they're all the same, so I'm only going to draw the all side chain, the difference. So first of all, is if R is H, this is alanine. Now be careful, we talk about the chiral, but alanine we have H and H, so it is an exception. The second one, if it's a methyl group CH3, this is, the first one is H is glycine.
Yes.
I forgot about this. Second one, CH is alanine. So glycine is an exception. It's not a chiral structure. Then we have CH, CH3, CH3. This is valine. Then we go here, we have CH, CH2, CH, and CH3. This is leucine. And then we have CH2, CH3, then we have CH3, sorry, this CH3 in the bottom. So that's CH2, CH3. This is called isoleucine. And the next one, I have to draw the whole thing because it's a circle. So we will have NH2, CH, COOH. Then we have CH2, CH2, and CH2, and it's connected to the NH2. This is a proline. And this one, some people say there's no side chain because it's connected. Okay, because this part, they say it is off. Okay, now, so this is first non-polar. Now we're going to draw something a little bit difficult. So then we will have the R is, so this is called phenolalanine. But the next one also have a chromatic structure, which is called this is tryptophan. And then we'll have one which is a little bit debatable, is SHCH2. This is a cysteine. Now I look at all the textbook. There is a 30% of the textbook put cysteine into the non-polar structure, But 70 and 80% of the textbook, they are categorized this one as a polar but non-charged structure. So lots of people will say it's up to you to decide it, whether it is non-polar or polar, because it depends the SH, the charges. And here I also want to mention the tryptophan. The NH here, it's an amide group. They have a little bit of we call polar characteristics, but because there's so many CH here, so they can be neutralized, their polar structure. So therefore, we still say the tryptophan is a non-polar. Okay, then we have, this is the one, the cysteine. Then we have a last one here, which is CH3S, CH2, and CH2. This is methionine. Okay, so this is a non-polar structure. Now the easiest way to remember these, the non-polar structure, if it is composed, the majority of them are CH. And the majority of their side chains are all CH. Therefore, we will say it is non-polar. If you added something there, then it will become a non-polar. Sorry, the non-polar, which means majority of them are CH, and the polar, which means they're going to have some other structure. So we're going to talk right now is a polar. The first one is uncharged, but it is hydrophilic, which means they like water. So the first one is CH2OH. This is serine. And then you have CH3 and OH. This is serine. And when you see the OH here, hydroxy bonds here, this is, some people say it is easy to be combined with the phosphates bonds, then have ester bond there. Okay, now for these two things we'll have CH2C. This is called asparagine. And then you have CH2, and C, NH2. This is glutamine. Okay, then the last one, which is very similar to the phenylalanine, but we have a hydroxy bond on the side. This is called tyrosine. Okay, so this is the polar uncharged. Then we will move to the polar charged. The polar charged, we have a two group. Number one is acidic, which is negatively charged. So this is easy to remember. You have asparagine here and the glutamine here. Then it is easily you think about, you're going to have CH2, CO negative. This is called aspartic acid. Or some people say it is called aspartate. And then here, glutamine, so you have CH2, C goes here. This is glutamic acids or glutamate. Then we're going to move on to the last three, which is basic. Okay, basic is positively charged. And the first one, this one is the most complicated one. So we have NH, this is charged, HN, and go here. We have CH2, this is called histidine. And then the second one we will mention all the time later on when we talk about the Maillard reaction. We have a long chain of CH2, CH2, and then have NH3 on the side. This is called a lysine. And the last one is we have CH2, CH2, CH2. Then we'll have NH in the middle. We have C, NH2, and NH2 plus. And this is called arginine. I do not know, I want to say this, it's just a joke for you remember. H, L, and A. When I was a student, the faculty who make a joke, how you understand about her leggers or basic? Okay, that's maybe 20 years ago. H, histidine. Leggers, this is lysine. This is arginine. Okay, what does basic mean? It's like they say the millennials, the young people in the college, they don't study very well, and they are always dressing very fashion and spending time for the Twitter and the Instagram. So they have this joke there, okay? Then they want you to remember because it's the last three. They think you're not gonna remember very well because undergraduate class, they want you to remember everything, the side chain. So the faculty has been making the jokes there, but I don't want to talk about this too much because it's like a little bit insulting of the undergraduates in the campus. Is that right? So I don't want to say that too much, but if you don't remember it, you can know these three. Okay, so we finished talk about the side chain. Then we want to mention something related to the general microbiology or maybe nutrition a little bit. So first of all, this phenylalanine. And Michelle, you are taking my class before. This phenylalanine, if we join it, that means it's C, this is NH2, then this is H and COOH. And remember, we have some of the bacteria could do deaminase, take on this amino group, so the left side will become phenylpyruvic acid. And we talk about the aerobic respiration and the phenylpyruvic acid. It's a modified of pyruvic acid. When you have a pyruvic acid, then it can go like fermentation or can go TCA cycle, citric acid cycle, and all kinds of the other cycles. The second one is that tryptophan. If we want to draw the tryptophan, and then we will be, some bacteria can use tryptophan. They have this structure right here. This structure right here is called indo. And there is one bacteria, everybody knows. Some people make a joke, even you failed general microbiology, you still remember that bacteria is called E. coli. And the E. coli is Indo-positive. So you put a Kovac reagent there, and then the Indo will show the cherry red color. So these are something we talked about before. Now, methionine, this one, you should remember our first transcript amino acid is tRNA, MET. If you still remember this, we have this structure of tRNA, and this is 5', 3', then here we'll have AUG, and the first code is AUG, that's tRNA, methionine, and this is for E-standard modes. If it's for bacteria, we call it a formula FMET. And of course, we know this is carrying the anti-codon, and then the codon, which will be the mRNA, messaging RNA. So this one I want to mention. Now, polling this one, later on we will mention it's a kind of like a component of a collagen. Then we have a hydroxyproline and the proline, that's the structure of the collagen. Then the basic units will be the gelatin. We'll talk about that later. Now goes here. We want to mention is this guy. Lots of people like it because they use it as a flavor concentrate. So you like a chicken flavor concentrate, when you make a soup, you add it. It's a major component is glutamine, but the nutrition scientist will tell you do not use that too much because
Go ahead, finish your thought.
don't use that too much because they think it's gonna be hurt you like the brain function later on if you use that too much.
You're not referring to monosodium glutamate, though.
No.
You're not referring to MSG.
Yeah. No, I'm not referring to that. I'm just basically talk about that. MSG is one of that. There is some, if you look at the ingredients, there's lots of them are MSG, but I just want to talk about that a little bit.
Yeah, I was just going to comment, Dr. Shen. I think the glutamic acid, the monosodium salt of glutamic acid is the MSG.
Okay, yes.
Yeah.
It's actually this one.
Yeah, actually, yeah.
It is that one.
So
they actually have salt.
So yeah, Na, so the monosodium salt of glutamic acid is MSG.
Yeah. That one. Okay. And histidine, this guy, and has a little bit, we call it anti-inflammation activity. That's a histidine. And this lysine we will mention. Because this is to the alpha carbon is 1, 2, 3, 4. Here we also call it epsilon. We call it epsilon amino acid, and this one is very easy to be reacted with the sugar, carbohydrates, and involved in a Maillard reaction. And the major components for lysine in a food product is milk powder. So that's why milk powder is easy to turn yellow if you store it in the room temperature for a long time. So we will mention this again. Okay, so here is the thing we mentioned about, briefly introduce some. Now we want to talk about several things. Number one, there is one concept you learned in your nutrition class, which is called essential amino acids. The essential amino acids, which means they not can be biosynthesized by yourself. It has to be taken from the diets. So, what are they? When I was students, they said there is 8 right now. There is 9 amino acids, which is essential amino acids. What are they? Valine, leucine, isoleucine, and we had phenylalanine, and tryptophan, and methionine. Then go here, we have serine, and right here we have lysine. So can you count 1, 2, 3, 4?
It's an arginine.
Yes, the arginine. Yes, where it is.
No, that's okay.
Last one. Okay. Now they also mentioned about there is 6 other amino acids, which is they call it's conditional essential amino acids. I think it's got alanine, proline, cysteine. And here we had a tyrosine. Another one is glutamine. Those 3 we call it limited or conditional amino acids, which means infant baby do not have an ability to digest them, and also we call it a metabolic disordered patients may need help for that. They cannot digest that. So that's essential amino acids. And also I want to mention, there is a standard to talk about the digestives of amino acids in the body, which is we call it a protein corrected amino acid digested score. And it is used by FDA and also by WHO and the FAO, the Food Agriculture Organization by the United Nations, they used to give a score of the amino of the food products for their containing the amino acid digested ability. So they gave egg is 100 score. They think egg is containing most of them.
Isn't histidine also?
I think this should be histidine.
Yes.
Yes, this should be histidine. Not that one, probably.
That one's the one that they said is conditional.
Yeah, conditioning. This
Yeah.
is conditioning. It should be histidine. Because I remember the histidine is that structure is kind of difficult, and that's an essential amino acid. So they give the egg a 100 score, and now in a developed country. In developed country, they usually using animal protein as a major nutrition energy resource in the developed country. In the developing country, like in Asian countries, they are using plant protein and as a major energy or nutrition resource, but they always missing some of the amino acids. For example, they use corn. The corn, they do not have lysine, and also the serine. And they also have another one, which is soybean protein. Soybean protein they use a lot in China as an energy nutrition resource, but soybean protein there is no mentioning there. So this is mentioned a couple of examples. So how they solve the problem? They are generate modified, genetically modified plant protein to genetically composed with most of the essential amino acids there if they're missing some. Okay, so this is amino acids we mentioned. Then one more thing I want to mention. Do you want to move the camera a little bit? Yeah, just cover the side. Thank you. Yeah, just cover that side. And we want to mention is so-called the PI of the amino acid. So what is that? So we mentioned about amino acid structure. So we have NH, C, H, R, and COOH. Now what is supposed to be? If it's at a neutral, a near neutral pH, we call it a near neutral pH. These will be charged. This will become NH3 is positively charged, and then this is become COO negative, which is negatively charged. But then they will go two directions. Is keep a balance. So on the left side, it could become NH3 R H and COO. This will be H and protonated. So we have a pKa1. And then on the right side, it could become NH2, and then go CR, and H become COOH. And this is called pKa2. And so what is pKa1 means? Which means a pH at which the protonated carboxy concentration equals the deprotonated concentration. And here the pKa2, which is indicated pH at which the NH3 plus the concentration is equals the NH2, which is pronated the amide group and the depronated amide group. So what gonna be the PI means? This PI means a pH at which the whole amino acids, there is no, or we say it's electrical neutralized or neutral. And this is important. We mentioned that if a muscle has been at the isoelectric neutral status, the pH, it will have a lowest ability of the water holding capacity, so not going to hold the water very well. Then in the POC, it will be showing the PSE characteristics. That is not good for the POC, so we mentioned that. So that's a PI. That pH is a PI. Now, how to calculate the PI? It depends. Your amino acid, the pKa value, pKa1, pKa2, and also the side chain. Because when you look at here, some of the amino acids, the side chain, which is charged, and at least the polar charged acid and the basic, but also some others, like let's say cysteine, and also the another one I think is asparagine. It's both of these two, and have a charge of the side chain, so they will be included into the calculation. So how to do that? So the PI, if it is number one, if it is non-polar and no charged, then the PI value is pKa1 and pKa2 and divided by 2. Second, if it is polar and acidic charged, then that will the PI will equals pKa1 add pKa3 and divided by 2. Number 3, if it is polar but basic, which just means it's positive charged, then that will be PI equals pKa2 add pKa3 and divided by 2. Because the side chain, there could be ionized. And I forgot to mention the whole thing for amino acid near neutral pH has positive and negative charge at the same time. We call it the Zeta ions. Okay, and then you also learned in your class, which is a biochemistry class, we could do is this type of the curve. The y-axis, x-axis is pH. That's from 1 to 13, so we can divide them into several. 2, 3, 4, 12, 13. Then the y-axis will be the equivalence of basic and acid ingredients. So the top will be, this is 0, 0.2, 0.4, 0.6, all the way go to 1.0. That will be equivalent of sodium hydroxide. And the bottom one, you have 0.2, 0.4, 0.6, 0.8, and you go 1.0. That will be the equivalent of HClO. And so what's going to be the curve looks like? The curve will be looks like this, and we call it a titration curve. And here the value is pKa2, and at the top is pKa1. And right in the middle, you see this area is very flat. This is PI. So this is something you learned in your biochemistry class, and you can review that. And here, I believe there is a table in the biochemistry class, you will do the calculation. Now, if I remember, most of the pKa1 is about 2.00. And the most of the pKa2 is about 9.00 to 10.00, somewhere around 9.2. Now, pKa3, it depends. It could be up to 13, 12, and it could be only like 2 or 6 or like 3, those small numbers. So that's the table for you to do the calculation. Okay, now here we finish talk about amino acid. Then we will talk about the protein structure. So the first, we need to know the protein composed by amino acid, mainly. There is a 4 different structure. Number 1 is primary structure. Now, primary structure, we usually call it is a number and sequence of amino acids. And how they composed. So let's give an example. If it's NH2, C, or glycine, let's say, and they have COOH, and then here when we draw carboxy group, we draw it like that. And then we have another amino acids. Let's say we add them together. NH2, C, let's say we have a alanine, H, and this COOH. Okay, this guy, we also draw it differently. Will be N, H, and H. So what's going to happen when they combine? They're going to lose the water. So water going to go because OH and H can become water. So the water will be released, and this is we call it a condensation reaction. This is a layman language. We call it a dehydration reaction, and it is not only for the protein, for the lipids and for the carbohydrates, they are all the same because these are the monopolymers and they become a large large polymers. So when they combine together, they will become NH2, CH, H goes here, CH goes here. So we have NH, CH3, H and COOH. And then we will say, right in the middle, CO and NH, this is, we call it a peptide chain. And then we have the one which is from here to here. This is, we call it a peptide unit. Now, the proteins, they could be composed by a different size of the amino acids. If they only have 2 amino acids combined, we call it a dimonomer. Or dipeptide, sorry, this should be dipeptide. It's called dipeptide. If there is a number is between 3 and 10, we call it an oligopeptide. You know what is oligo means? It means just a few. So that's 3 to 10. Now most of the proteins, they are amino acids, is very large. It's averagely about 300 to like 1,000. So this is we call it, it's a polypeptide. Poly, which means many, okay, many kinds of. So this is the first, is a primary structure, is number and the sequence of the amino acids. But I want to make sure it is very important. Even there is a little bit of change of this number and the sequence of the amino acids. At the end of the day, the protein structure or the function could be changed. Now, number two, we want to talk about is secondary structure. Secondary structure, we call it's a localized, regular conformation of protein. Now, what are the good examples? You know that, which is alpha helixy and beta, we call it a pleated sheet. And the last one is called random coil. And the alpha helix, what it looks like. And I use a chalk pen draw like this. Not thick enough, some here. Okay, it's like this. Okay, alpha helix, they are connected right here. Actually, is a C, and this is H, N, and they connected here with hydrogen bonds. And this one term is about 0.54 nanometer, composed by 2.6 amino acids. Now, who is the person who found this? Is a great American scientist. Is Pauling. You learned about that. And we mentioned a couple of times, the Pauling has another contribution, which is electronegativity and the sp3 hybrids. And the Pauling, when he find this, he win not for this. He find this. And at the time, in 1950s, early 1950s, people want to find the structure of the genetic information, which is DNA. So he's getting very close to that. He has a small paper published to think about the DNA should be very similar to this. However, he did not cross the finish line. He's washing on the creeks. They published the paper. They said it's the most beautiful 20th century article in the Nature. So you know that if we do this, this is DNA double helix. Is that right? So alpha helix of the protein is very similar to that. Now, what is a beta pleated sheet looks like? This is kind of like this. This is not thick enough, so like this. And here is go here. And then here will be a circle. Okay, that's a beta pleated sheet. Now, random coil will be just the sum of the coil. They say there's a A, then there is a B. And sometimes, if it's composed by the cysteine, then they will be combined with SS bonds. That is a disulfur bonds. And the disulfur bonds is a strong bond. It's stronger than the hydrogen bonds and the other non-covalent bonds. So that's a secondary structure. And the third one we call it a tertiary structure. So what is that? That is usually we say it's a 3D structure of alpha helixy beta sheet, which is folded, or we say compacted into a globular structure. So what that means, if you have a unit like that, then inside this unit you have a alpha helixy, so you have a beta pleated sheet, you have these type of the random coil, and they are all connected, maybe connected with hydrogen bonds or other non-covalent bonds, or sometimes is biosulfur bonds. So that's a tertiary structure. Now the last one is a cautery structure. Cautery structure, which means assembly of, we say, protein molecular and polypeptides into a subunit. So they gave an example, which is a hemoglobin. Hemoglobin, you have these four subunits, which is at the top is beta, and the bottom is alpha. They can carry on the oxygen during the blood transport in the body. And the cautery structure, we also can differentiate protein into two different types, which is fibrous and also globular. And this is usually non-solvent, and basically is a structure. And this one is solved, is a solvent, is dissolved in the water, and basically it's an enzyme. A good example is an enzyme. Okay, so these are the structure for the proteins. Now we want to mention some of the examples of the proteins. The example for the proteins is number one is a simple protein. And the simple protein, just give you some example. Number one is albumin. And this one is usually widely exists in the egg, and it's dissolved in water without salt, or neutral water without salt. Number two is globulin, and this one is in a soybean, for example. We have globulin, soybean globulin, and we have conglycinin. It's beta-conglycinin, and also have a beta-glycinin. In muscle foods, we have myosin and actin. We'll talk about that cross-link, which is have to be break down during the meat tenderizing.